Okay, I have checked on everything. The microphone is on, notifications are off, although you know how that goes. And I've got everything ready for you here. So we can talk about from gene to protein. We had been talking about, let's go with green here. No, purple. We had been talking about DNA replication. And there's a notification already. All right, DNA replication is just making a copy of the DNA, and I just needed to add a little detail for you. As you know, the DNA is coiled, and helicase unwinds the DNA and basically opens it up. So I've got to open it up a lot here. Helicase. And so you'll have all these G's, A's, T's, and C's here. And if this is our 3' end up here, our 5' end will be down here. And we'll have all these G's, A's, T's, and C's down here. I won't write them in this time because I already did this. I just want to point out that DNA polymerase is the enzyme that will add complementary bases starting with the new 5' end first, in this case heading towards the replication fork. But the deal is that if this yellow dot is DNA polymerase here, it's got to actually bind to a strand of DNA in order to be able to add these bases. But the deal with DNA polymerase is that if this is DNA polymerase, I'll write DNA pol here, DNA polymerase can't bind to single strands of DNA. So DNA polymerase can't bind to single-stranded DNA. I'll just put ssDNA for single-stranded DNA. That's actually a very common abbreviation. It needs 2 strands to latch onto. So what the heck? How's it going to do that? The cell synthesizes a very small piece of, let's use red for this. A small piece of RNA to bind to the DNA that's complementary to the DNA, so that DNA polymerase has 2 strands to bind to. So actually, let me complete this a little bit here. I'll just add a couple more bases. These are where the bases would be. First base, second base. And so the cell will synthesize a small piece of RNA. So this is RNA. And this is actually called an RNA primer. A primer is something that gets something going, like if you're going to prime the pump. People have to prime a pump to get it going sometimes, so like a water pump, something that pumps up water from, say, a deep-driven well. You have to prime it. You have to get it started. So an RNA primer, it's a small piece of RNA that gets things started. So, and it will be synthesized here from 5' to 3'. Now DNA polymerase, oh my gosh, I'm getting all of these notifications from Instagram. Apparently, I put up a controversial post on Instagram thanking people for wearing masks, and a bunch of anti-maskers came out. And I had to handle that because, I forgot this is the internet. Okay, that aside, we need to go back to the yellow. Isn't that what I had for DNA polymerase? Now DNA polymerase can bind to this whole thing and start synthesizing DNA, adding complementary bases, starting with the 5' end, because the RNA primer gives DNA polymerase 2 strands, a second strand to latch onto. Okay, so you've got the replication fork here, and the leading strand is heading in that direction. For the other strand, yep, the cell has to make a little RNA primer there. That gives DNA polymerase something to latch onto, and it'll move down this way. And, you know, when it reaches here, it'll add the new 5' end first, heading away from the replication fork. So this is the lagging strand, because when helicase unwinds this and unzips this back this way a little more, we're going to have a new replication fork. So it's a kind of a funky thing to explain in words, right? But when you get helicase on unwinding this whole thing, you'll be able to see all the spots where, you know, RNA polymerase had to make little primers, but you can see all the spots where the old replication fork used to be, because the leading strand will be smooth, but the lagging strand is going to have all of these Okazaki fragments. Okazaki fragments right there. And so there are going to be all of these little gaps here, right? We're going to have a 3' end here, 5', 3', 5', just tons of little gaps right there. And who's going to close up those gaps? Well, a couple of things need to happen. First of all, the cell's got to get those the RNA bits out of there. Got to get those RNA primers out of there. So let's use a different color. Let's use green. Another DNA polymerase will actually cut out the RNA primers, fill them back in. So let's write this down. A different DNA polymerase cuts out the RNA primers, because the cell's not okay with having little bits of RNA inside its DNA, inside the chromosome. Doesn't like that. So that another DNA polymerase cuts out those RNA primers and fills it in with DNA. And then DNA ligase will seal up those gaps. So then DNA ligase joins the fragments to each other. There we go. Okay, so just a few more details for DNA replication. Make sure you can explain in words how we get a leading strand and a lagging strand. And it's all because DNA polymerase can only add the new 5' end first. That's what it's all about. And because DNA is anti-parallel, you're going to have a 3' end here and a 5' end there, so it can only add the new 5' end first. One heading towards the replication fork and another one heading away from it, but helicase can't unzip the whole chromosome that fast all at once. You can see that these are the spots where the old replication fork was. I mean, like, how's helicase just going to unzip the whole thing all at once before these DNA polymerase molecules can get in there and do their job? So that is DNA replication. So this whole thing that we've been talking about, DNA is copied, and the copy is called RNA, and then RNA is read, and the ribosome builds a protein from it. But I said, oh yeah, we got to do this part here. This part here, that's DNA replication. Now we're going to focus on this stuff up here. So there we go. I love this little thing. Love this stylus. All right, so this whole process of DNA being copied, or rather transcribed into RNA, because that's transcription. And then the RNA being read and translated, so this is translation, translated into protein. This whole process we basically call gene expression. Gene expression. Okay, the gene is in the DNA, and the RNA is the copy of the gene, and the protein is the gene product. We say that when a protein is made from the gene, that the gene has been expressed. So that's what we mean by the term gene expression. Okie doke. So, first thing we got to talk about is transcription. So let's talk about the transcription process. The transcription process begins with DNA, a gene, and makes a copy of it into RNA. But I just want to talk about how there are different types of RNAs. So there are, well, actually here, why don't we go right here? Why? It's so funny how it's doing this. It keeps reverting to this file that I didn't even open. Okay, gene expression. Not all RNA is translated into protein. There are different types of RNA, and they can finish up then. Now, most RNAs are going to be this first variety, messenger RNA. We call that the transcript. Messenger RNAs actually are translated into proteins. Ribosomal RNAs become part of the ribosome. Transfer RNAs are part of the translation process, but they don't get translated into protein. And I'll talk about this interference RNA in just a moment. But first, let's go back here and pick another color. How about navy blue? All right, so there are different types of RNA. Sorry, different types of RNA. I didn't mean to put an M in there. My brain gets ahead of my mouth. There is messenger RNA, or also known as mRNA. We're never going to write the word messenger RNA out again. Messenger RNA is translated into protein. This is the protein information. So it has the protein information, also known as, the protein information are known as codons. And we'll get to what those are in a couple minutes. So it has the protein information, is translated into protein. And we'll come back and take a look at what that looks like. There is ribosomal RNA. Oops. Be right back. Dropped my pen. Okay, there is ribosomal RNA. Or rRNA, which is kind of a funny thing to say. rRNA, it's like you're a pirate or something. rRNA. These pieces of RNA are actually part of the protein. Part of the ribosome. And I can't spell. They are part of the ribosome. So those pieces of RNA that Carl Woese sequenced to determine the 3-domain system, this is hearkening back to like just the first week of classes, eukarya and bacteria and archaea domains. He sequenced things like what's called the 6S ribosomal RNA, things like that. So that's just an example. So these are actually part of the ribosome. They do not have protein information. So don't have protein info, and they don't get translated into protein. They play a huge role in translation into protein, but they themselves don't have the protein info. Another type of RNA that is involved with translation into protein but doesn't have the protein information itself is called the transfer RNA. Or can you see it coming? tRNA. tRNA does not get translated into protein. But it plays a role in translation into protein in that it brings the amino acids to the ribosome. And tRNAs have something that we call an anti-codon. So each one will have a specific anti-codon. So back up here, messenger RNA has the codons. tRNA has the anti-codons. Yes, they are related to each other. And just quickly, I wanted to mention that this is so weird, but whatever. I was having trouble with my Google Docs with PowerPoint on this opening up, and just doesn't want to do it. So I'm leaving this in Dropbox, and it's just not liking this interface. This interference RNA by Fire and Mellow. It sounds like an 80s band, Fire and Mellow, like Peaches and Herb. I'm gonna need yellow here. Interference RNA. These are RNAs that play a role in turning genes on and off. So they do some part of, this is not a pen, I want this, part of gene regulation. Turning genes on and off. I don't know what the Fire person's first name is, but Mello, this is a dude named Craig Mello, and he was over at UMass Worcester, at the UMass Medical School in Worcester years ago. I don't know if he's still there or not, and he did some of his work there as well. So I just thought I would mention that there are other RNAs other than the big three: messenger RNA, ribosomal RNA, and tRNA. Delete that. All right, I just want to quickly talk about what, get back to this in a minute. I'm going to focus on this transfer RNA for a little bit because it's got a funky shape. All right, and to talk about this funky shape that it gets, like let's say we've got this piece of RNA here. So it's going to have a 5' end and a 3' end. And let's see, you know that RNA is single-stranded, but let's say we've got a sequence here that something's like CCC, AAC, U, not U, ACC, oh, how about A, U, G, U, G. All right, you know that C's over here on the left are complementary to G's. And you know that that A's are complementary to T's in DNA or U's into RNA. So I'm looking at these 3 C's on the left over here. That's going to be complementary to 3 G's. Are there any spots with 3 G's on this piece of RNA? Huh? You see it? Oh, look, there are 3 G's down here. Can you see that they're going to be attracted to each other and want to form hydrogen bonds? Like that. And like, what would this AAC be complementary to? Wouldn't it be complementary to a UUG? You see anything where there's a UUG thing? Oh, look, here is a UUG. Oh, look, they're going to be complementary to each other. And these AAAs are going to be complementary to a UUU, yeah? Where am I going with this? This whole thing is going to fold in half. Like if you have your hands, if you put your 2 wrists together, put your hands together and then fold your hands back so that, you know, your elbow, hand, here's your hand, and you've got your hand back here. The other one like this. There's your other hand. Out like that. Now put your hands together. Put your hands together. This thing is going to fold up like you just folded your hands. This works so much easier when I'm just standing in front of you, but I think adding a personal video and doing a podcast for you all is not as effective as actually being able to run on the board. But this thing here is going to fold up like a little hairpin. 5' and 3' like that. And there's, you know, a C, C, C over here, and there's a G, G, G over here. And they're going to be attracted to each other. What am I getting at? The tRNA has that going on. And it folds up into a funky shape that, interestingly enough, looks like the letter T. Today's lecture is brought to you by the letter T. I feel like just getting rid of that one file that's in there. All right, so I'm just going ahead. Aha, here we go. The letter T. This is a diagram of a transfer RNA right here. So let's just take a little picture of it. And let me see. All along here, where I'm drawing, maybe if I did it in black. This is the piece of RNA. So all the nucleotides are in there. This doesn't show up so well on the screenshot, does it? Okay, that's the piece of RNA. And these dots here, those represent hydrogen bonds. So these are these are complementary regions. So like if there's a C over here, there's a G over here. I can't write a C in there and a G in there. There's just not enough space for me right now. And these bubbles, well, the thing's got to there's got to be some sort of hairpin turn. I mean, ergonomically, I would still say these are non-complementary regions. It'll be like C, 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 A, or something like that. But like if there's a C here, there's going to be a G here. That kind of thing. So it folds in on itself, and it has complementary regions to itself. Now check out this spot down here where it says anticodon. And here we've got a C, a U, and a G. That's going to become very important later on when we talk about translation into protein. But I just want you to hold on to that idea that tRNAs have this thing called an anticodon at the bottom. And check out at the top. There is an amino acid up here. In this case, it's aspartic acid. So earlier when I said tRNAs bring the amino acid to the ribosome. This is what I'm talking about. They actually have an amino acid covalently bonded. That's why the word covalent bond is right there. Covalently bonded to them up at the top. So, but there's no protein info in here anywhere. All right, so let's get back to transcription. We'll need this in just a second. Well, let's just take a look at this right here. Okay, up here at the top in red and green, we've got a piece of double-stranded DNA. And they're complementary to each other. There is a region. There's a region of DNA before the protein info called a promoter. Sometimes we call it the promoter region. So, I'll come back to that. Here we go. So let's look at transcription. All right. So I'm not going to draw 2 strands of DNA and write all that in, all those G's, A's, T's, and C's out there. That's going to take way too much time. What we tend to do in biology is to just draw one line and say, here we go, here's the DNA. Period. You just got to imagine that there are G's, A's, T's, and C's in there. And when we want to say that there is a region of DNA, that there is a gene in a region, we tend to put just, better pick a color than that. We tend to just put a little box on it. So we just draw this little box on there and say this is the gene. I don't know, gene X, something like that. In front of the gene, or what we call downstream, what we call upstream of the gene, there is an area that we call the promoter, or the promoter region. So, by the way, if this is upstream, this is downstream. The promoter region is a series of a particular sequence of DNA nucleotides that RNA polymerase recognizes and binds to. So RNA polymerase is the name of the game here. It's the name of the, what color do I want to pick for RNA polymerase? Let's go with orange. RNA polymerase is the name of the enzyme that's going to add complementary nucleotides to one of the strands of DNA in the gene. So the promoter region has, let's just define this better first. It's an area upstream of a gene that RNA polymerase binds to. Upstream of a gene that RNA polymerase, and I'm just going to write RNA pol, I'm not going to write the word polymerase out, we'll abbreviate, that RNA polymerase binds to. Now, a promoter region, it's a specific sequence, it has specific sequences. And it varies from promoter to promoter, but usually promoters have a lot of T's and A's. In fact, it's often called a ta-ta box. And yes, I know that the word ta-ta refers to something else. So, for example, October is Breast Cancer Awareness Month, and occasionally, like at HCC when we were there in past Octobers, there would be signs of, raising awareness for breast cancer and encouraging women to have mammograms if they are due for one, and encouraging people to fundraise for it. And there was one sign at HCC, gosh, it was right down by Fronahue where the pod is, and it said, "Save the Tatas." And I was like, why are people talking about Tata boxes and promoters? Are they in danger? What's going on? I'm like, oh, it's October. They mean a different type of Tata. but that's what we call them, ta-ta boxes. So RNA polymerase will bind to this specific region of DNA. So if this is RNA polymerase, I still think this is a little too thick of a pencil. There we go, RNA polymerase. And it has RNA polymerase has a part of it called the sigma factor. So the sigma factor, and this here is a sigma, it's, it looks like a circle with a little hat on it or something. The sigma factor of RNA polymerase is actually what recognizes the promoter region. So, what's interesting about this is that RNA polymerase, it has a sigma factor that will bind to the DNA, to one strand of it, but RNA polymerase doesn't, it actually can bind to one strand of DNA, so you don't need any RNA primers in this. So there are no RNA primers in this. So RNA polymerase is able to open up the DNA and it begins adding new complementary nucleotides to one strand of the DNA, beginning with the 5' end first. So, similar to DNA replication, the new RNA polymerase makes the new 5' end first. So, RNA polymerase binds to the promoter. It melts the DNA, separates the strands into single strands, and it's able to bind to it. And RNA polymerase adds complementary nucleotides, but they're the RNA flavor. They're going to be U's instead of T's. There's going to be ribose in there instead of deoxyribose. So that's what I mean by RNA flavor. Adds complementary nucleotides to one of the DNA strands. And which strand it does, it's going to have to add to the 3' end of the DNA, because RNA polymerase always builds the new 5' end first. So, transcription proceeds 5' to 3', just like DNA replication, 5' to 3'. And what it will do is that, starting somewhere before the gene, it will move along this way and it'll start adding complementary nucleotides, making the new 5' end first. And it will usually find some sort of signal. Let me go actually just a little farther. Come back here. There's some sort of factor here that signals RNA polymerase to stop. So now we'll have this little piece of RNA, and it's got the protein information. So it's the messenger RNA right there. Let's take a look at this. Is just going to keep happening. And I'm sorry, it's just happening too many times for me to edit out. All right, let's take a look at this little graphic that I've got here for you. Easier if we take a screenshot. All right, so there is some sort of signal where transcription begins. That's our transcriptional start site right there. And let's just enlarge this so that you can see that the new 5' end is made first. Right there. And a 3' end here. Notice that this strand, RNA strand here in purple, is going to match this red strand up here. So we've got only there are going to be U's instead of T's. So we have CACUU. Up here there's CACTT. GGUUCU, GGTTCT. So it's essentially going to match this red strand. And in your book, yes, this strand is called the plus strand, and this green strand down here is called the minus strand. Don't worry about that. I'm not going to hold you responsible for that. All right, so we'll come back to this in just a second. I want to talk about a few differences with transcription between eukaryotes and prokaryotes. With eukaryotes, this piece of RNA up here, and actually I'm going to get rid of that. FYI, no, I won't do it that way. Let's see. In eukaryotes, if you have this little piece of RNA, the 5' end was made first, 3' end down here. Oftentimes, in eukaryotes, there is some extra processing up to this RNA, and certain pieces of it will be cut out. So I'm just going to put that there. And the cell will cut it out. Right there. So the green part, that's missing now. This green part here is called an intron. And eukaryotic mRNAs often have introns. Prokaryotes don't have introns. Prokaryotic mRNAs don't have introns. So no introns. So on that running list that you have, of differences between eukaryotes and prokaryotes, this is another one to add there. I think the last thing we added there was probably an asymmetrical cytoplasmic membrane for eukaryotes and a symmetrical cytoplasmic membrane for prokaryotes. Now you got something else. There's a difference in their transcription. The mRNAs get processed a lot in eukaryotes, but not in prokaryotes. There are introns in eukaryotes, no introns in prokaryotic RNA. Something else that is quite interesting. We'll just erase this stuff right here because I don't want to go too far away from my list. Is that if you've got in eukaryotes, like let's say this is a big piece of chromosome and you've got a gene here and a gene here and a gene here. So gene X, gene Y. Can you see it coming? Gene Z. In eukaryotes, there will be a promoter in front of upstream of each gene. And so you'll get a little messenger RNA for each gene. This does not sound earth-shattering to you. So in eukaryotes, you get one messenger RNA per gene. This is called monocistronic. A cistron refers to an area where there's a gene. I don't make this stuff up. I just let you know. But prokaryotes are not the same. No, no. Prokaryotic cells will often have one promoter for a bunch of genes. So let's see. Yeah. Get to that in a second. And so it will have one long messenger RNA. 5 prime. Oh, got our 3 prime end here. With protein information for 3 different genes. So there's actually one promoter and one messenger RNA for several genes. So more than one promoter and messenger RNA. Oops. For a gene. So more than one. This is greater than sign. Over here, let's on the left, let's revise this a little bit. One messenger RNA and one promoter per gene. That's monocistronic. With prokaryotes, you often have one promoter and one messenger RNA for several genes. So this is polycistronic. Polycistronic versus monocistronic. This chapter has lots of new vocabulary words for you. So there are some differences there. Alrighty. Come back to Dropbox, and of course we have to go back to this weird sizing chart thing. You must be like, what the heck is this? All right, so you get processing in eukaryotes that you don't get in prokaryotes. All right, so now we've got this, what's called a mature transcript. If we're talking eukaryotes, the transcript is already mature in bacteria and other prokaryotes, so we're good there. And the cell is ready to start translation. I do want to just go through this slide one more time. Okay, transcription uses RNA polymerase, no primers necessary, got that. It adds a complementary single strand of RNA to one of the strands of DNA. New nucleotides are added, 5' to 3'. I want to point out that either strand of the DNA can be transcribed, and it's the orientation of the promoter that tells RNA polymerase which direction to go in. So for example, if we've got this piece of DNA, let's change color now, we need to do this. If we've got this piece of DNA, and there's a gene here on one strand, there could actually be a gene on the other strand with a promoter region. I'm going to spell the word promoter backwards. Can I do this? We did it. Going that way, and a promoter going this way. It's the orientation of the promoter that tells RNA polymerase, hey, there's a gene right here, bind here and start adding to the strand so that you can add the 5' end first, 3' end here, and RNA polymerase binding here and making a little strand this way. So it's the promoter that decides. And yes, you could actually have an overlapping gene this way even. Really cool. Cool stuff. And of course, we have to go back to this weird thing. I have a feeling if I deleted it, it would still show up. All right, so transcription, orientation of the promoter tells RNA which direction to transcribe. All right, so to summarize, differences between eukaryotes and prokaryotes. The messenger RNAs are modified. They have introns in eukaryotes. Prokaryotes, nope, no introns, no modifications. Eukaryotes are monocistronic. One gene, one promoter, one messenger RNA, one promoter per gene. Prokaryotes, you often have one promoter and one messenger RNA for several genes. So you'll have several genes on one piece of messenger RNA. This last little bullet is important. Translation can't happen right away in eukaryotes because think about where the DNA is. In eukaryotes, the DNA is inside the nucleus, right? The chromosomes don't come outside the nucleus to anytime genes need to be transcribed. They stay in there. The only time that chromosomes come outside of the nucleus is when that cell's dividing by mitosis or meiosis and the nuclear membrane disintegrates. So transcription occurs in the nucleus. And then the messenger RNAs have to get out of the nucleus first before they can be translated into protein. But prokaryotes don't have that issue because they don't have their DNA in a nucleus. The DNA is right here. Transcription begins in the cytoplasm. And the ribosomes are all out here in the cytoplasm. Yep. So transcription can begin right away. Sometimes it can even happen, like a messenger RNA won't even be finished transcribing yet when the ribosome will latch on to the 5' end of it and start translating it into protein. So that's kind of cool. Kind of cool. All right, so that gets us into translation into protein. And so let's finish up chapter 7 and talk about translation. Translation into protein. So we're going from messenger RNA, which is often, by the way, called the transcript, which means copy, basically, into building a protein from what the messenger RNA says. So to do that, you need a few things. Well, you've got your gene. Let's back up. Let's talk about the DNA. And let's just deal with a monocistronic gene for right now. So this is, gene X. And there's a promoter here. And let's say RNA polymerase bound to the promoter, and it built a transcript from this. Here we go. Give it shorter to 3'. And it's going to have, U's, G's, A's, and C's in there. So, G, A, U, C, A, C, A, U, G, C, A, C, blah, blah, blah. And if this is a eukaryotic cell, this little messenger RNA has to get out of the nucleus and out into the cytoplasm where there are ribosomes that can look at the order of all these G's, A's, U's, and C's and build a protein from that. So let's say that it got out of the nucleus. There we are. Now our little messenger RNA is available to the ribosome to build a protein from it. So here's how it works. And you know what? This is going to be too small. I need to make a bigger piece of messenger RNA. So let's say I'm just going to write the same thing all over again right here. Blah, blah, blah. 3' end's going to be way down here. What did I have here? G, A, U. This doesn't matter all that much. C, A, C, A, U, G, C, A, C. I'm just making this up. G, U, A, U, G, G for Ugg boots. G, C, U, A, U, something like that. And these are just all the little nucleotides sitting off here. And I'm just going to erase all this stuff down here. Okay. So the ribosome actually has to bind somewhere near the 5' end. And it binds at an area called the, wait for it, ribosomal binding site. Isn't it nice when something in science is actually named for what it is? It's like, okay, the ribosomal binding site sounds like some place where the ribosome will actually bind. And that's what it does. So somewhere here, and it's just a particular sequence. It's not GAUC or something like that. It's much larger than that, but because I can't draw something that's, 200 bases long, 200 nucleotides long, we're simplifying things a little bit. So you've got a ribosomal binding site, and that's what the ribosome binds to. But the ribosome is made up of two parts. The ribosome is made up of a small ribosomal subunit and a large ribosomal subunit. I'm not going to write the word ribosomal. You can all hear me with that. And if we're talking prokaryotes, the small subunit is the 30S ribosome, and the large subunit is the 50S ribosome. And you know that put together, because we talked about this way back when, put together they make a total of a 70S ribosome. Yes, we know that 30 plus 50 is 80, but when you put them together, it's a size thing. It's not a weight thing. So, the whole ribosome doesn't bind all at once. The small subunit binds to the ribosomal binding site first. So the small subunit binds first. So I'm just going to draw it like this and call this the 30S ribosome right there. So it binds. And then the small subunit moves along the messenger RNA until it finds the first AUG. So let's just write this down. Binds first, and it moves along the messenger RNA until it reaches the first AUG. And the AUG, the first AUG, is called the start codon. The start codon. Okay, the word start means, well, to start, but the word codon has a different meaning, and it just means, what is a codon? A codon is just 3 nucleotides in a row. 3 nucleotides in a row. So an A, a U, and a G is a codon, and the first AUG is a start codon. There are other codons that have other letters, like GUC is a codon as well, but the start codon is AUG. So let's scroll up here. So it moves along, we'll erase that, until it finds the first AUG. Oh, and look, here's one. Here's an AUG. So the 30S ribosome has moved there. The first AUG tells the ribosome, this is the start codon. This is where the protein information begins. So what happens then? It's really cool. Oh, we're going to need this room up here. Let me get rid of that. We need another color. Okay, so the 30S ribosome moves along and binds to the ribosomal binding site, moves along the messenger RNA until it reaches the first AUG. Then it allows a transfer RNA to come through and bind to the codon. So remember the tRNAs look all funky like the letter T way back. Well, I didn't show you that. I guess I didn't draw it. Good thing. It was the thing that looked like, this. With an amino acid up here and an anticodon letters down here. That's what that looks like. So I'm going to draw this tRNA. It looks like this. And it's anticodon, and I happen to know that this one's anticodon is going to be UAC. Do you notice that there is a relationship between UAC and AUG? And if you said, oh, they're complementary sequences, you are right. So they actually hydrogen bond to each other. You know that A's and U's will form 2 hydrogen bonds to each other? And G's and C's, 3. That's what holds the tRNA in place. Now up at the top here, remember there's going to be an amino acid that's bonded to it, except, okay, there are 20 different amino acids. I happen to know that this particular transfer RNA has a methionine up at the top, covalently bonded to it. So this looks kind of like a hot mess, but that's not. All right, once that happens, the large ribosomal subunit, the 50S subunit, can bind to the whole thing. And it's pretty big, so I'm just going to draw it like that. And the whole thing, here, I should have done a better job drawing the 30S, it actually is more like that. So this is the 50S ribosome, and this is the 30S ribosome here. I think it kind of looks like the Great and Powerful Oz or something like that, or maybe Squidward from SpongeBob. I don't know. I gotta amuse myself here too, people. Okay, so what happens is that when the 50S binds, it opens up a space for the next codon. And a codon, remember, is 3 nucleotides in a row. So C, A, C, that's a codon right there. So a second transfer RNA will come in and bind, and oh my gosh, this is going to be a hot mess. They're going to have their arms all around each other like this. This one's anticodon is going to be a G, U, G. Alrighty, and there's going to be an amino acid up here. Now, I just happen to know that methionine is always going to be the amino acid that's up there because I got that memorized. I don't know what this one happens to be, and I don't expect you to memorize that either. I expect you to learn how to use what's called a genetic decoder. Let's see if I have that. Oh, I do not have that open right now. I have it listed. Okie doke. This is a decoder. And let me take a screenshot of it to show you how it works. Now, the way that I knew that it was going to be methionine here is that I use this decoder for it. First up, when you use this, and I have uploaded it to the Moodle site, it's called the genetic decoder. I want you to write down, look up the codons, not the anticodons. If you look up the codons, you'll get it right. If you look up the anticodons, you're going to get it wrong. So we're looking up the codons. So what we're looking up is, for example, AUG. The first thing that you do is that you look up the first letter and you start from the middle of this wheel. So A. The second letter, you move out to the second circle, U. The last letter, you move out to the outermost circle with the letters, G. And look, it says methionine right there. Methionine. It's a little bit fuzzy because I'm zooming in a lot here, but you can print this out if you like. Alrighty. So what was the next codon? Oh, we're done with that. Got to delete the screenshot before I can toggle. Next one is I'm looking up is CAC. So I'm going to look up CAC. And by the way, don't look up GUG. Don't look up GUG. We're looking up CAC because that's the codons. AUG is the first codon. CAC is the next codon. GUA is going to be the third codon. UGG, etc., etc. So we are looking up CAC now, not GUG. So we're looking up CAC, start in the middle, C, oh say can you see, A, and then C, and it is histidine. Histidine is what the next one will be, histidine. I'm just going to write hist because I can't fit histidine in there. Okay, so to make a protein, we got to join these two amino acids here. And if you remember back to chapter 2, was that chapter 2? When we talked about the dehydration reaction that joins two amino acids to form a growing polypeptide chain. Ooh, let's see if I can draw that. Let's see, can I draw this? We've got an amine end here, a central carbon there, the carboxyl group on one, and methionine. I don't know what the R group is for methionine. I don't remember. And I sure don't know what it is for histidine off the top of my head. Oops, not that. Whatever the R group is, remember an enzyme comes along, pulls out a water molecule, and then bonds these two together, and that's called a peptide bond. So dehydration reaction, that is what is happening up here, right here. We're doing a little dehydration reaction. Now, this transfer RNA over here, the first one that came in, can leave the building. It can go away. And as it leaves, that's actually called the indexing step. I don't really care that you know what that's called, but I'm going to give you a video to watch, and they refer to it as the indexing step. When this first transfer RNA leaves the building, leaves the ribosome itself will completely shift. This causes it to shift one codon to the right. So let's do that and see if I can do this. Get that out of there. That's there. And I gotta draw that back in. Erase this. And now the ribosome is going to move here and over here. I can draw that better. There we go. Clean this up just a little bit. All right. Okay. I am getting really tired of drawing these tRNAs. How about now that we know what they look like? Oh, dear, I'm running low on battery. Now that we know what they look like, I'm just going to draw them like a letter T, and I'm going to put an amino acid up here at the top, and I'll write the anticodon down here. So if GUA is the codon, GUA is the codon, the anticodon is going to be CAU. Okay, are we going to look up CAU or are we going to look up GUA? If you said we're going to look up CAU, you are in some trouble. You got to look up the codons, not the anticodon. So let's look up GUA. G is in the center, so right here, G. And then where's the U? The U off to the left, and an A. So it is valine. Valine is the winner. There we go. Oh, and if we looked up CAU, can you see? Oops, come on. AU, we're going to get histidine again? That's wrong. We have to look up GUA, and that's going to be valine. So I'm going to write valine up here. I'll just write valine. Enzyme comes along and does that neat little dehydration reaction, and now we have 3 amino acids joined to each other. So now this transfer RNA can head out of Dodge now, and the indexing step can occur again. So it's going to leave the building. Bye-bye, tRNA. And the ribosome is going to move 3 clicks, or 3 nucleotides, or basically 1 codon to the right again. Something like that. All right, so everybody, what is our next codon? It's UGG. That's our next codon right there. So that tRNA is going to come in. I'm getting a little tired of my color coding, but you know, I'm trying to do this right. It's going to be ACC, but we're not going to look up ACC. We're going to look up UGG. Let's see what UGG does for us. UGG. UGG. Tryptophan. Remember, you start in the middle, the U right here, and then move to the second letter, the next circle out, G, that's right here in the middle here, and then G, outer circle, tryptophan. Oops. Come back. Here we go. So it's tryptophan. I'm just going to write trip right there. Enzyme comes along, joins them, forms a peptide bond by doing the dehydration reaction. I probably don't need to keep moving the ribosome for you. Maybe we could just keep going with this, because now all we have to do is look up the codons, don't we? Let's add a few more here just to give us a little more practice. So our next codon is going to be GCU, AUC, and let's say UGA. Oh dear, UGA. I may have to come back and finish this lecture later, or find my power cord somewhere. All right, so the next one, so let's see. We did AUG, that means methionine. CAC, that means histidine. GUA, that means, I can't read my writing, valine. UGG, tryptophan, our next one is GCU. So we're looking up GCU. So G, G, and then the C in the middle, and then out in the yellow lined circle, U, alanine. So I'll write ALA. And a dehydration reaction joins them as well. How about AUC? Oops, no, come on. I am trying to make this work for all of us. Now I can't remember what it was. It was AUC. Can I see that without? Yes, AUC. So A from the center, then U, moving the next one out, C, and it looks like, isoleucine. That's how you say that funky word, isoleucine. Isoleucine right down here. So we started with A, and then U, and then C, isoleucine. I'll just write isoleu. And they get joined by another dehydration reaction, blah, blah, blah. Can you see this is just going to go on forever and ever? Let's look up the next one, UGA. Let's look up UGA. I'll take a screenshot of this because that works better. Okay, so U right here, G, A. Stop? What? What is up with that? What does this stop mean? What that stop means is that there is no corresponding tRNA that will be complementary to the UGA. There is no tRNA carrying an amino acid for the codon UGA. UAA. It is a stop codon. Notice also that UAA and UAG are all stop codons as well. That means that's a signal to the ribosome. There is no more protein information here. You're done. So there is no tRNA that binds to that. There's none. I'm not even going to draw that. There we go. So that's it. So that's the protein. So when the ribosome comes to that, it actually has help coming off the messenger RNA. Those are called like protein releasing factors, terminating factors. I don't really care that you know the names of those, but again, I'm assigning a video for you all to watch, a YouTube video, and they're going to mention that stuff in there. So let's see. So yeah, that's the process. So you should be able to look at any piece of messenger RNA, 5 prime, blah, 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 all the way down to 3 prime. If I give you, G, A, C, U, A, C, A, C, C, A, U, G, C, A, C, 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 G, U, A, C, A, something like that, you need to be able to predict what the order of the amino acids is going to be. Now remember, this was transcribed from the DNA. So this information originated from the gene, but now it just has U's instead of T's here, and the ribosome is going to bind to this end here and look for what? Ooh, it's going to move along until it finds the first AUG, isn't it? This is the start codon. So you need to be able to look up AUG and say, oh, well, that's going to be a methionine. And then you need to look up CAC. I forget what that was. What was CAC? We found it before, didn't we? Yeah, histidine. That you look that up and it's a histidine. And then you need to look up CCG and then UAA. Ooh, that's a stop codon, actually. So you need to be able to do that. So meanwhile, I am assigning you a video to watch. And I just put the link up on Moodle. It's a YouTube link. It was made in 1970 something or other. I don't know. I want to say 1977. Sometime in the 70s. So the quality is really crappy. And it was done at Stanford University in California, and it was a college-wide project that a bunch of students worked on. A play of sorts, a dramatization, that's the word I'm looking for. They created a dramatization of translation into protein. And all I can say is that at the time that this was made, I think LSD was still legal in California, so you can take that with a grain of salt there, but you're going to see some weird stuff in this. But it's a really helpful video. But I did want to point out that because it's a dramatization and they do like an interpretive dance, it's absolutely freaking hilarious. They wrote a poem in the style of a poem by Lewis Carroll. I'm not sure how you spell Lewis Carroll. I think that's wrong. I think it has 2 R's. Maybe 2 R's and 2 L's. I don't know. I don't know the spelling. But Lewis Carroll is an author who wrote Alice Through the Looking Glass and What She Saw There. So which is actually kind of a weird psychedelic kind of book itself. But in Through the Looking Glass, he wrote a poem called The Jabberwock, or the Jabberwocky, can't remember. And he tried to make it sound like this epic Old English poem like Beowulf or something. And it started off with, 'twas brillig, and the slithy toads did wire and gambol in the wabe. And he made up all these words to make it sound like Old English using words that were lost. Why am I saying this? Because this video that I have asked you to watch actually does a parody of this poem. And it's interesting. Like I said, And I think this was actually made when LSD was still legal in California. So enjoy that, and we can chat about this on Monday or Tuesday, whichever time you have class. So I hope you have a great day, had a great weekend, and we'll talk soon. So bye-bye.